Today we'll discuss about testing of drugs on human volunteers. Just observe the heading, Testing of drugs on human volunteers. During your research, if you prepare any compound, whether it may be antimicrobial or anti-diabetic or anti-cancer, before releasing the drug into market, we need to conduct tests on human volunteers. So that's why the heading is Testing of drugs. Drug may be your compound or any blood related product or anything related to gene therapy. So first we need to do trial on human volunteers. So human subject research, basically it involves a systematic investigation. It can be either research or clinically oriented. Basically involves the use of human volunteers. Clinical trials includes various experiments and the drug maybe it's a drug or medical device or biological such as vaccine, blood product or gene therapy. So if you observe this slide, first point, if you observe on first point, we require human volunteer. Second point, we need to conduct clinical trial. And third point, the treatment could be a drug or we may be testing a medical device or any vaccine. Vaccine also, if you compare with the present scenario, even the prepared vaccine first has to be tested on human volunteer. So today's we are studying the topic called testing of drugs on human volunteers. Observe the slide again. So first we require human volunteer. We need to conduct clinical trial and we need to test our drug or any medical device or vaccine. If you observe the next slide, the research may be related to various fields. It may be a basic biology or clinical medicine, nursing, psychology, sociology, political science, or anthropology. Any human research. To test a drug on human volunteers, we need to follow certain guidelines. And the first guideline was declared at the end of Second World War and it is referred as Nuremberg Code. The voluntary consent of human subject is absolutely essential. If you conduct any research, if you want to release any product, then the consent of human subject. First you need to test the product on human volunteer. His or her consent is required. So, for example, you are doing any plant research. You have extracted a compound and you can declare the compound is anti-diabetic or anti-cancer. In lab, you can perform several experiments and you can tell the given compound is anti-diabetic. But to prove it is anti-diabetic, you need to do some in vivo assays. You can take ethical clearance, you can conduct tests on animals. Again, if it proves anti-diabetic, then you need to move further on human volunteers. If you observe the slide properly, the guideline basically what we need to follow is the voluntary consent of human subject. If you want to test any drug, the consent of human is required. And directly you cannot test a drug on human. First you need to conduct laboratory assays, then take ethical clearance, conduct animal experiments. After animal experimentation, with a knowledge of natural history of the disease or any other problem under study, you can perform the results on or you can test your drug on human volunteer. What are the guidelines we require? So just consider now we are testing a drug on a human volunteer. We need to follow certain guidelines. Study involves the research of unproven drug. We don't know about the drug efficacy. It may be vaccine or any product. The purpose of trial should be informed to patient or human volunteer, and time duration should be discussed with the human volunteer. The risks associated with study and which organ is being experimented should be informed. Possible risks or discomforts should be discussed with the human volunteer. Other procedures or treatments that might be advantageous to the participant, but we need to discuss. And also compensation, medical treatments, it might be advantageous to the human volunteer and it has to be discussed. And about the participants' rights to question about the clinical trial. And participation is always voluntary and he can quit the study anytime. So, if you observe the slide properly, before testing a drug on human volunteer, we need to seek his consent. Informed consent of patient is required. Time should be discussed. Risk associated with the treatment should be discussed. Compensation, medical treatment has to be discussed. And to conduct trials, there are 4 phases. Phase 1, Phase 2, Phase 3, Phase 4. So the degree of risk to be taken should never exceed that determined by humanitarian importance of the problem. Basically, you are testing a drug on human volunteer. So your experiment should contain 4 phases. About phase 1. Phase 1 usually consists of small number of healthy volunteers. It may be around 20 to 80 people. Mainly it deals on how drug is metabolized, how it is excreted, and also to identify acute side effects. For example, you have discovered a drug. So the drug should be metabolized, it should be excreted, and it has to be with less side effects. I mean, efficacy should be more. Once phase 1 is cleared, next we'll move on to second level. Second phase includes more number of participants who have the disease condition and that the product potentially could treat. It includes a group of 100 to 500 people. If these trials indicate drug may be effective and risks are acceptable, then it moves to next level. Just observe the slide properly. We are conducting a testing of drug on human volunteer. First phase, we have injected the drug, and if we get accurate results, we'll move on to phase 2 with more number of people. If phase 2 is cleared, we'll move on, that is phase 3. Usually phase 3 drug is studied in large number of people. It is carried among 1000 to 3000 people, which includes product effectiveness, and again we need to monitor side effects and again comparison of the product effect to a standard treatment. For example, you have extracted one anti-cholesterol compound from fungi, and Biocon will release normal commercial product called lovastatin, just for an example. If you compare your drug with the standard drug, if you do the comparison and if you get a better results, then we'll move on to fourth level. So basically your drug should compare the effect to a standard treatment. And once phase 3 is cleared, we'll move on to next level. Basically launching of a product. To find out more about the treatment, long-time risks, benefits, optimal use, or to test the product in different populations of people, different population, different age group, etc. So basically our topic is we are testing a drug on human volunteer. First thing, we require informed consent. We need to follow few guidelines, and then our clinical trials includes 4 levels: phase 1, phase 2, phase 3, phase 4. The guidelines basically depends on the age, sex, type and stage of disease, which is very important. Treatment history is very important, and other medical conditions. The test involves people with particular illness or condition to be studied, and few may seek healthy volunteers also. Next question comes to our mind: who are these clinical trial teams, which may include doctors, nurses, healthcare professionals, researchers, etc. They are required to check the health of the participant at the beginning of the trial and assess whether that person is eligible to participate and who agree to participate. So while during the the trials includes, we need to check the health of participant at the beginning of the trial, and we have to assess whether that person is eligible to participate and who. Usually it is done at hospitals, research centers around the country, and all the clinical trials are monitored with the help of protocol maps and usually under the guidance of FDA. Who reviews the trials? The entire drug testing trials are reviewed by Institutional Review Board, and main purpose of review team is to ensure appropriate steps are taken to protect the rights and welfare of participants. If one such risk are found, research will not be approved by IRB. And usually this IRB team composed of at least 5 members from all healthcare professionals. Conclusion. So, the heading of topic is testing of drugs on human volunteers. Heading only conveys the meaning. You are testing a drug on human volunteer. The first and foremost condition is you need to take informed consent from the volunteer. And usually it includes laboratory phase. Whenever you do the research, first we need to do in vitro assays. Inside the lab, we need to prove whether the compound is anti-cancer, anti-diabetic or anti-cholesterol or whatever. And then we need to take ethical clearance and we need to perform this test on animals, albino rats or anything. In vivo assays we need to do. And after that, we need to do testing of drugs on human volunteers only under FDA guidelines. If you observe the current slide, the third point, that is testing of drugs on human volunteers, usually lies in the third phase. First you need to do research work in the lab, then you need to do trial using animals under ethical guidelines. You need to take ethical clearance, then only you can perform animal experiments. And then the main trial, that is testing of drug on human volunteer. So clinical trial basically involves a human experiment to study the drug efficacy and safety of a new principle, new active compound or a new drug. And in exam, how you need to remember is, if you get this question, that is testing of drugs on human volunteers, first you need to write about the phases, phase 1, phase 2, phase 3, phase 4, etc. And also you need to mention FDA guidelines. So main important thing you need to remember under this topic is, once you are testing the drug on human volunteer, informed consent, FDA guidelines and clinical trial phases, that is phase 1 to 4. At last, you can mention these points, that is laboratory phase, animal phase, and then on human, and ultimately the product will be released into market. And this topic is very similar to the present scenario, that is the preparation of vaccine and testing of drug on human volunteer. Thank you.